Okay, everyone, this is the video lecture for chapter 17. Um, I'll probably do this chapter in two separate parts, I imagine. The second part might be a little bit shorter than the first part, but that's okay. So, chapter 17 is on species, or more specifically, what's often referred to as the species question. You might think it's relatively easy to define what a species is or is not. In some cases, it is pretty straightforward, but in a lot of cases, it's not so easy to define individuals as members of the same species or as separate species, or to make distinctions between separate species. And that's kind of what this chapter gets at a little bit. So up to this point, we've discussed mostly microevolution, small-scale genetic changes over really short time frames, maybe one generation to the next, for example. Now, when we take all that microevolutionary change that we talked about as a result of selection, drift, migration, mutation, and all that, and we add it up or consider it over long time frames, then we're dealing with macroevolution, okay, large scale evolutionary change, and this typically does take large amounts of time. Speciation is typically thought of as a macroevolutionary process. So speciation is the process whereby one species maybe splits into two or more species, okay? We get descendant species from an ancestor species. It's a result of microevolution, but it's the end result of all that microevolution added up over long periods of time, which we think of as macroevolution. That's why we discuss speciation as being a macroevolutionary process, okay? So, speciation then is the origin of two species from one. It's also often referred to as cladogenesis. All right. So I'll get this slide here in just a second. Speciation links evolution at the population level to the evolution of taxonomic diversity. It bridges the gap between microevolution and macroevolution. All right. So cladogenesis is also often referred to as branching evolution. Branching evolution or cladogenesis can preserve at least some of the history of evolutionary change in a clade because intermediate character states can be retained in extant species, currently existing species. An analysis of these features is how phylogenetic trees are actually built or constructed. Contrast that with anagenesis, though, where we have change within a species, where one species morphs or evolves into a new species over time. That process of anagenesis erases the record of changes leading to the present form or the present species, although you may find fossil record evidence of some of that change, but you don't see the change or any evidence of it in the modern species that evolved from the ancestor. Excuse me. Okay, so an important question here is, what are species? Is it possible to define species in many ways? It actually is. And there are multiple different definitions for species. Some of these definitions are useful in certain situations, others may not be. It just depends on the situation, as we'll see. Carolus Linnaeus, who developed our modern classification system back in the early 1700s, define species entirely on morphological grounds, so outward physical appearance, body form. This is what's called the typological species concept. Okay, typological species concept. Let me write that down here on the front here real quick. Typological species concept. Okay, here, or in this species definition, individuals are considered members of a single species if they sufficiently conform to the type specimen, which is assumed to be representative of the species. So there's a type specimen in a museum somewhere, and it's recognized as the standard for that species. And anytime a new individual that's thought possibly to be a member of that species is found, it's brought in and compared. And if it sufficiently matches up with a type specimen, then we can consider it the same species. Now, you might imagine that might be somewhat subjective depending on the person and who's doing the comparing. And it is actually very subjective. The thing about typological concepts like that is that they don't take into account variation within a species, because you're constantly comparing everything to a single type specimen. So, if an individual seems to possess a mix of characters from two distinct species, how do you classify it typologically? And that's what we're dealing with this example. So, here we have on the left the common European raven, okay? Two closely related species to that are Corvus coronae and Corvus cornix. So this is the carrion crow and this is the hooded crow, all right? Now, in Europe, you typically find coronae, the carrion crow, in the western half of the continent, and the hooded crow in the eastern half. And there's a narrow hybrid zone between them where the two species actually do, or two forms actually do interbreed, and you get hybrids. All right. Now, the thing is that physically, these two birds do actually look very different. So if you see one from over here in Spain, that would be a carrion crow, and you see another one from, let's say, Yugoslavia, or what used to be Yugoslavia, a hooded crow, it might be fairly easy to define them as separate species. But in the hybrid zone, where they do actually hybridize with one another, you see individuals who are a nice mixture of both characters. And what do you do in that case, especially if you're relying on the typological species concept to define what is and what is not a species? My point in showing you this is that there are cases where the typological species concept just cannot be applied, even remotely objectively. And so maybe we need a different definition for how we define species because of the variation that can be present within a species or even within closely related species. Maybe these two species are better classified as subspecies of one another. But again, how do you make that call? Well, that brings us to what's called the biological species concept, okay? The biological species concept was introduced by Ernst Mayr in 1942, and Mayr basically defined species as groups of actually or potentially interbreeding populations which are reproductively isolated from other such groups. What Mayr was doing was defining species based on their reproductive capacity. In a nutshell, he said that two individuals are members of the same species if they can successfully reproduce, which means if they can produce fertile, viable offspring. If they can do that, according to the biological species concept, or the BSC as we call it, then they're classified as the same species. If two individuals can get together and mate, but they can't produce fertile or viable offspring that don't survive or offspring that cannot reproduce themselves, then those two individual parents are not actually members of the same species under the BSC. The nice thing about the BSC is that it's more objective. It places sort of a black and white criterion on the decision as to whether or not individuals are members of the same species or not, and that is, can they successfully reproduce? Basically, it defines species as protected gene pools, isolated gene pools that don't mix with one another. Okay. So the important aspect here then is reproductive isolation, which limits gene flow among populations. Gene flow does not have to be non-existent, though. There can be a small amount of gene flow between distinct species, hybridization, as we saw with the crows in the previous example. All right. So the biological species concept emphasizes the process through which species come into existence. The BSC is needed, it's necessary because it's known that morphological variation between species, or among individuals rather, is not a sufficient basis for defining species. All right, so what kind of observations support that? Well, first of all, there's considerable variation that exists among interbreeding individuals in a single population. Think about the different human races, for example, and how much morphological variation we see there. In some species of fish, you have what are called sneaker males and dominant males. And the dominant males look very different, extremely different from the sneaker males, but they are all members of the same species, although if you were looking at them and didn't know anything about them, you might well suspect they're different species because the two types of males do look very different. That's just a form of what we call polymorphism, differences in form, or multiple forms within the same species. we can't call these different individuals different species, even though they look quite different morphologically, because they are capable of, or they exist in the same gene pool, essentially. All right, we see geographic variation as well. Individuals differ across a spectrum on a geographic basis, such that the presence of intermediate forms between extreme forms suggests interbreeding between those extreme forms. We have what are called sibling species. You have reproductively isolated populations that are essentially identical morphologically in terms of outward physical appearance, but they may have distinct ecological, behavioral, or genetic chromosomal differences that actually prevents them from mating with one another. All right, so the nice thing again about the biological species concept is that it's the most objective species definition that we have, but there are some problems or limitations, not problems, I should say limitations to it. First of all, it's restricted only to organisms that reproduce sexually. What do you do about an asexual species where it doesn't require two parents to reproduce? You can't test the BSC that way because the BSC requires that you have two individuals that can successfully reproduce with one another. Asexual species doesn't require two parents, just one. The BSC is also essentially limited to modern organisms, extant species, since it's impossible to know whether or not a modern individual could have mated with one that lived several million years ago. And when you look at species that are only present in the fossil record, they're now extinct, you can't tell anything about whether or not they could have reproduced with one another just by looking at the fossilized bones. So there, maybe we have to look at what's called the paleontological species concept, which is based more on bone anatomy, essentially. All right. Okay. So the term species basically has two meanings. One refers to reproductive capability. That's the biological species concept. And the other is simply as a taxonomic category. Asexual species are species in the taxonomic sense. We can define them taxonomically, but they're not biological species in the sense of the biological species concept. So a lot of bacteria, for example, sort of fall into that category. Now, we also have borderline cases. You can have populations that are referred to as parapatric. Okay, parapatric. This just means that their distributions are adjacent to one another. So maybe you have one geographic range here of one species and you have a second geographic range there, and their distributions, their ranges actually bump into one another. These two are referred to as parapatric. And a lot of times what you get there is a hybrid zone between them. And the crows over here are a great example. So Cornyx on this side, Coroni on this side, they are parapatric and they have a hybrid zone in between them. And so you do get these hybrids sometimes forming in these hybrid zones, and often they're called semi-species. There's a lot of different names that are thrown out there to describe these kinds of situations. But, you know, it just depends on the particular situation we're looking at. Okay. Now, in a lot of cases, we have situations where you have two populations whose ranges actually overlap with one another. So maybe we have, for example, one geographic range here and a second one there, and we have this area in the middle where they actually overlap. That is referred to as sympatric. Okay, sympatric. You have an area of each, or of one range rather, that overlaps in some area of the other, and you can get hybrids forming in there because there individuals of the different gene pools, or populations if you will, can actually come into contact and maybe interbreed. That kind of hybridization, sympatric, is actually very common in plants. Okay. All right, so good example of that, sympatric hybridization. Here you see some examples of hybridization or hybrids. So we have Quercus grisea, which is the gray oak, and we have Quercus gambelii, which is the Gambel's oak. And these are our southwestern species. So you find them in, for example, let's see, Texas, I believe New Mexico, Arizona, parts of Colorado. But their ranges broadly overlap in those four states. And on the fringes, in the parts of the ranges that don't overlap, you either get Quercus grisea or you get Quercus gambelii, But in the ranges where they overlap as sympatric zones, you can get a variety of different hybrids, which you see here, for example. Do you call these different species? Do you call them the same species here and different species when you compare those guys? Same species, different species. You see where the subjectivity can come into play here. So the point is that this idea of species is not, in most cases, very cut and dried. It can be quite complicated, we should say. Okay, so if we're talking about the biological species concept, which for the most part, as biologists we are, because that's the one that is the most objective, so we apply it when we can, realizing we can't always apply it. If we're talking about the biological species concept, we need to talk about what are called barriers to gene flow. We know that gene flow between populations can reduce genetic differences between them. It's like mixing white paint and red paint, right? They homogenize, basically. In order then for enough differences or genetic differences to accumulate between two populations to allow them to speciate and actually become reproductively isolated groups or new species, gene flow must occur only at low levels or be entirely non-existent. So, according to the biological species concept, speciation consists of the evolution of barriers to gene flow. Evolution of barriers that prevent gene flow and therefore allow species to become different over time through new mutations and adaptation to unique environments. So, barriers to gene flow can be categorized in two ways. We can talk about what are called prezygotic, prezygotic barriers, and post-zygotic barriers. So, zygotic, think zygote, fertilized egg, right? So, prezygotic barriers are those that prevent formation of a zygote. Post-zygotic barriers are barriers that kick in after formation of a zygote, so post-zygotic, all right? Now, most prezygotic barriers have their effect before a mating attempt. So most of these different categories of prezygotic actually prevent mating attempts between members of distinct species, okay? There are some prezygotic barriers, however, that while they don't prevent a mating attempt, they can prevent zygote formation should mating occur. So they're referred to as post-mating prezygotic barriers. They happen after mating but before formation of a zygote, right? So let's take a look at some of these. And sorry I forgot to put in blank slides here for this presentation to write on, so I'm just going to have to write on some of these figures, okay? So let's look at pre-mating barriers. First of all, we have what's called ecological isolation, okay? So this refers to various things in the ecology of the species that actually keep them separate from one another. So we have habitat isolation. Here, two species may live in different habitats and simply never come into contact with one another. If they never encounter one another, how can they possibly mate, right? Well, that's not true in plants because pollen can be wind-driven and that sort of thing, but we're talking about animals here for the most part. So we have habitat isolation. We can also have temporal isolation. So it could be that you have two species that do live in the same area, they do encounter each other on a daily basis, but maybe they don't come into reproductive age or season at the same time. Maybe members of one species, males of one species are ready to mate in May, and maybe females of the other species are ready to mate in maybe March or April. And so their reproductive cycles are out of sync, basically. That would prevent them from forming zygotes, wouldn't it? All right. Then we have behavioral isolation as a second category. So we had ecological isolation first, and within that we had habitat and temporal isolation. Now, as a second type of pre-mating barrier, we have behavioral isolation. Again, another way to prevent mating. So this is really important in sympatric species that come into contact with one another. The specific system of mate recognition of a species prevents mating between members of different species. So females, usually, not always, but usually will only be receptive to males capable of displaying the correct signals, displays, songs, that sort of thing, reproductive behaviors, courting behaviors. Only males of the same species of the female will be capable of actually doing the correct songs or displays or courtship dances or whatever. a male of a different species is not going to be able to do the correct displays to attract the female of a different species. All right, good example of that. Here we have species of damselflies, and here you see sonograms of their calls, the male calls, okay? So we have one species here. Look at the frequency of the male call, okay, very distinctive call. For a different species, you can see from the sonogram just how different that male call would sound. And we have even a third one here that would be just as different from the other two as they are from each other. So if this is the, or maybe this is the caddisfly, if this is the call that the male has to make to attract a female, a male of this species does this call, which means a female of that species is going to be attracted by that call, not this call and certainly not that call. So these are behavioral features in terms of the mating calls of males that act to keep the respective gene pools of those species separate from one another to prevent hybridization. All right. These morphologically distinct, identical species, they're actually lacewings, use different songs to distinguish between individuals of the different species. Okay. All right. That's just an example, an auditory example. What about visual signals? Well, here we have a great example of visual signals. These are often very important as a means of behavioral isolation. Females rely heavily on what are called secondary sexual characteristics of males to discern their own species from others. So very often, especially the birds, for example, males of the species are the ones that are very brightly colored, flashy, showy, that sort of thing, because again, they're trying to attract females. And we talked in the previous chapter about how females very often cue in on the overall health of a male and therefore his potential as a mate or a good mate via his physical characteristics. Bright coloration, that kind of thing is a good way for a female to discern that. So you can imagine a female of this species is looking for that in the male. She's not looking for this, and vice versa, right? So these very obvious visual cues or visual signals are keys to the females, typically the species, as to, what males are the ones they should mate with and which ones they should not. All right, now, that's animals, but plants rely on morphological differences in their flowers, for example, to attract different pollinators. In this way, a plant can actually increase the likelihood that pollen from another conspecific, which is a member of the same species, will actually pollinate its eggs. So the idea here is that if you have a flowering plant that depends on pollinators to visit it, to get a nectar meal, and then to take some pollen away and then visit another plant, that plant that just had a pollinator visit it wants that pollinator then to go to another plant of the same species, not a different species, because if it goes to a different species, the pollen that it gave to that pollinator might be wasted. So what we have then are morphological features, differences in these flowers to attract different pollinators. So these flowers, for example, tend to be bee pollinated, whereas these tend to be bird pollinated. There are other categories of flowering plants that are pollinated by moths, for example. So the point is that if these individual species of flowering plants can limit the number, or the type rather, of pollinators that come to them, bees only or birds only or whatever, then they increase the chances that when that pollinator leaves that plant, that pollinator will go to another plant of the same species. And that's the idea. Okay, so those are prezygotic barriers, okay, pre-reproductive or pre-mating prezygotic barriers. They all prevent a mating attempt, essentially. Now, what happens if a mating attempt between two different species occurs? Well, then we can have what are called post-mating prezygotic barriers kick in. So again, we have a mating attempt in this case, but still no zygote formed. So it's a post-mating prezygotic barrier that prevents that zygote from forming. All right. You can think of these barriers that kick in after a mating attempt as sort of fallback mechanisms to prevent formation of a hybrid zygote. So let's say a female, for example, may terminate copulation if the male's genitalia do not result in the expected tactile stimulation that the female's expecting. That might be a cue that he's not the same species as she is. Okay. Good example of that. Here we have some examples of genitalia of different species of male fruit fly. So we have a lot of morphological variability in these lobes here, for example, okay? And, well, for lack of a better way to put it, the parts have to sort of fit together, essentially, to make things work. And if the pieces of the puzzle don't fit together, then you're probably not going to get a zygote formed. So you might have a mating attempt in these cases where maybe a male of this species tries to copulate with a female of species A up here, but if the morphology is not right, then there would probably be no sperm injected or left in the female's reproductive tract, or if they are, then you can have other barriers that we'll talk about later on kick in. But the point is, you may have a mating attempt here, but still no zygote form. So they are post-mating prezygotic barriers. All right. Also, there are some cases where we have sperm from a male of a different species that is sometimes unable to compete with sperm from a male of the same species. So those sperm from a different species are referred to as the, or the male rather of a different species is referred to as the heterospecific male. Hetero meaning other or different, so different species male. And the sperm of the heterospecific male is often unable to compete with sperm from a male of the same species, the conspecific male, if both of those sperm types happen to be present at an ovum, an egg, at the same time. If both are present at the egg at the same time, and the egg is fertilized by sperm of the conspecific male, that is known as conspecific sperm precedence. So conspecific sperm precedence. All right. Okay. So there are cases where you can get sperm from males of different species at or present at an egg at the same time. More often than not, though, the conspecific male sperm will actually be able to fertilize that. This is referred to as gametic isolation. Okay, gametic isolation. This again is post-mating prezygotic barrier. It occurs when sperm and egg are simply incapable of uniting. This is common in a lot of species of marine invertebrates that release eggs and sperm simultaneously into large clouds of gametes over a few nights of the month in the spring when the moon phases are right. You have just these clouds of gametes from potentially hundreds of different species of invertebrates on a coral reef, let's say, that all end up in the water as a gamete soup. Well, what prevents sperm from species A from fertilizing eggs of species B when they bump into them, which they do? Well, it's usually molecular differences, especially in terms of surface proteins of the different gametes. So failure to unite in that case is due to differences in cell surface proteins in the sperm and the egg. It can be subtle amino acid sequence differences in the proteins among the species that prevents heterospecific gametes from uniting. All right, so again, that's a post-mating prezygotic barrier, okay? All right. Well, I think that we'll stop there for this one, and we'll pick up with post-zygotic barriers in the second half of this chapter.